Hello, and welcome to Genetic Engineering, Season 1, Episode 6992. Today we'll be learning about CRISPR and Cas9 gene editing technique. Joining us on the show today are two special guests, Ms. Dr. Smith and Dr. Liang. Dr. Liang's from China and Mr. Smith's from Canada.
Doesn't matter.
God! Okay, let's give a warm welcome to them. Hello, Mr. Smith. Nice to meet you. Nice to meet you. I'm Dr. Smith. Can you please explain to me what genetic engineering is and how it works and how it functions?
Sure. So basically, genetic engineering involves taking the genome of one organism and altering it in some way, so you can cut out certain genes or mix and match genes with different organisms, all to produce a new desired trait.
Can you give me an idea of what type of work you're doing?
Yeah. In China, we are on the verge of being the first in the world to inject people with cells modified using the CRISPR-Cas9 gene editing technique. A clinical trial received approval from the hospital board on July 6th. This trial will enroll patients who have lung cancer and for whom chemo and radiation therapy failed. We will extract immune cells called T cells from the blood of the patients, then use Cas9 technology, which pairs a molecular guide able to identify specific gene sequences on a chromosome with an enzyme that can snip the chromosome at that spot to knock out the gene in the cells. The gene encodes for a protein called PD-1 that normally acts as a check on the cell's capacity to launch an immune response to prevent it from attacking healthy cells. Gene editing cells will then be multiplied in the lab and reintroduced into the patient's bloodstream. Engineered cells will circulate and hopefully home in on the cancer.
Dr. Smith, is there any negative side effects to this process?
Well, you know, in new technologies, there's always unknown factors, things to be worried about. For example, in this particular example, there's the potential for the gene going into the wrong place, which could mess up many of the normal cell processes. It could turn the immune system of the organism against the healthy cells. There's all sorts of things that could go wrong, but we're hopeful.
Yeah, we hope that this trial will have a positive effect.
Sounds very interesting.
So Sophie, how do you feel being a patient in this job?
Well, the doctors have been very supportive and caring, and I'm really, really looking forward to the positive results.
Okay, so the technology we're using is called CRISPR, which is an acronym for something that I'm not going to define. It involves a Cas9 protein and an RNA strand. This technology was first found in bacteria. A virus would attack the bacteria and try and inject its DNA into the bacteria's DNA. If the bacteria survives, it'll steal some of the virus's DNA and store it inside CRISPR. So if there was another viral infection, what the bacteria would do was it would send Cas9 to go and check all the DNA in the cell, and if it found a match, it would, with the RNA, if it found a match, the Cas9 would cut the DNA off, removing it from the genome. Now what's great about this is it can be programmable, so you can give Cas9 any set of RNA, and it'll cut out any genes, any chromosomes within the genome. So you can remove traits, but you can also add traits if you put in donor DNA, which the cell will use to replace the missing gene.